biology department at Eastern with us today. This is the last week of our spring semester, but as you can all tell, it's really the beginning. If we had a summer semester, it's really the beginning of summer. So we appreciate Barbara coming to us to talk about viruses and vaccines, which as we're coming out of our confinement is really, really an interesting topic. And she's going to talk about the past and the future of pandemics. Barbara?
Yeah, thank you very much for that kind introduction. And thank you so much for inviting me. It's always a pleasure and a real treat to speak to this crowd. So what I want to do is just start off with a timeline. Hold on, let me see. A timeline of COVID-19. So in December of 2019, China had a report of an outbreak, a cluster of pneumonia, and this was in Wuhan. Later, we came to know that this is where the coronavirus that we're familiar with now that causes COVID-19 infection, originated from. Within about 10 days, the WHO, World Health Organization, disseminated guidance on how to detect this virus, how to test for it, and importantly, how to deter the spread. And we're all familiar with these behaviors in terms of social distancing, wearing masks, washing your hands, don't go out if you don't have to, that type of stuff. This red arrow here is indicating that on January 12th, there was a real milestone that was hit. And this is setting the foundation for all of the good science and vaccines that we have today. 12 days after this cluster of pneumonia was identified, the genetic sequence of the virus that causes it was publicly shared. And it's that genetic sequence that was the key to making all of the vaccines that we have today. 2 months later, COVID-19 was then designated as a global pandemic like none that we have ever seen in our lifetimes. So what I want to do is try to bring a little bit of information to this group about viruses in general, then coronaviruses, and a couple of sections on the vaccines that we have, how they work, and how are they developed. And when I talk of the vaccines, I'm going to speak specifically of the ones for SARS-CoV-2, which is the virus that causes the infection COVID-19. So before I begin, though, I want to ask you, what comes to mind when you hear the term virus? What do you think of?
Spreadable disease.
Spreadable disease? Infection?
Not able to reproduce except by hijacking other cells.
Right. They need a host in order to make more viruses. They can't do it by themselves. Yeah, excellent. Anything else? Okay, so some people, sometimes maybe you'd think of a virus. Okay, well, that's how you get the common cold. Maybe a cold sore is caused by a virus. You probably didn't think about global pandemic until recently. You probably didn't think about viruses or microorganisms that can infect and kill bacteria. And maybe you didn't think about how abundant they are in our oceans. So viruses are actually spectacularly diverse in the types of viruses that they have and that are around, and in their organization, in their genetics, and then how they can interact with hosts. So they're found virtually everywhere on Earth, and they can infect any organism, from the most minuscule bacteria to the largest organism like an elephant. But they can also infect plants or insects and many other different types of animals. So, and they're found in virtually every ecosystem. You can find them in marine habitats, in freshwater, and in terrestrial habitats. They're in the air that we breathe. They're in the ocean when we swim there. They're in the ocean when we don't swim there. But the point is that they're all around us. And because they're so small, we can't see them. And it makes us somewhat unaware of them because we can't see them. So they are exceptionally small, which I hope led you to ask yourself, well, how small are they? Okay, well, I could give you a measurement on how small they are, but that doesn't mean much to most people. So here's a question I'm going to ask of you. How many viruses could you fit on the head of a pin? And you're just going to answer this to yourself. Could it be 5,000 viruses, 50,000, 50 million, 500 million, or too many to count? So choose one of those answers. So if you chose number 4, 500 million virus particles could actually fit on the head of a pin. That gives you an idea of how small they were. So if you answered number 4, you would be correct. If you can't count up to 500 million and you answered number 5, too many to count, you're also correct. Okay, so viruses come in many different shapes and versions. And because of these different structures, it gives us the idea that, okay, they're not all the same. There's thousands of different types of viruses. So if you look here, we see they could be cylindrical viruses. They could have many different sides to them. This is called a polyhedral virus. An example of a polyhedral virus is something called adenovirus. And I'm going to ask you to just put that in the back of your mind because that adenovirus is going to come up later in the talk. There are spherical viruses. You probably recognize from what you've seen in the media and on the news that this is the shape of coronaviruses. And some of them, they look like spaceships. This would be a type of virus called a bacteriophage. And these viruses are the ones that can infect and kill bacteria. So there's vast diversity in viruses. Okay, so I would not be doing my job if I didn't make this point. Viruses are from Venus and bacteria are from Mars. Well, this is just another way of saying viruses are different from bacteria. It's really important for us to understand the difference between these two and for your doctor and medicine to understand the importance, because you don't want to treat a viral infection with the drugs that we would use for a bacterial infection. And we have something, a global health crisis that seems to be on the back burner because of the pandemic, but it's still with us. And that is antibiotic resistance. Antibiotic resistance refers to the fact that bacteria, sometimes even in the presence of antibiotics, which are drugs that are meant to kill them, they can still thrive and grow. The implications for us is that we have fewer and fewer and fewer antibiotics that can treat bacterial infections. So people are dying from, I'm going to call them simple, they're not really simple, but simple bacterial infections because we're running out of antibiotics. I've talked within the last 8 years, I've given 2 talks on this subject. It's very close to my heart. I've talked to the United Nations in New York City twice. And I brought a program to Eastern Connecticut State University where undergraduate students are trying to discover new sources of antibiotics to help us with this antibiotic global crisis. So they do research to try to find new antibiotics that are isolated from soil bacteria. And this is part of a global program. There's thousands of students around the world that participate in this program, but it sort of brings to life this whole global health crisis of antibiotic resistance. So again,
Could you explain a little bit how that antibiotic resistance happens? Does the bacteria mutate or it be able to, get around the antibiotics?
so you could have an entire talk just on that, but simply, I think David, was that you that? Yep. So it is that the bacteria mutate, and now the drugs that used to kill them and prevent them from making more and more bacteria, they're no longer effective. So the bacteria just live and grow, even though we're trying to give drugs to them to prevent it. Now, how all of that happens is it's very complex, but I'd be happy to come back and talk to you on another occasion. Okay, so the point here is viruses are different from bacteria. And some of these points you guys already brought up, like viruses can't survive or make more viruses, which is what replicate means. They can't make more themselves by themselves. They need a host. So they have to infect a cell and then hijack the machinery within that cell to make more viruses. Bacteria, they're standalone organisms. They can survive and make many more bacteria all by themselves. In terms of size, bacteria are much larger than viruses. Bacteria, if you have a bacterial infection, you would treat it with antibiotics or vaccines. Whereas if you had a viral infection and you use antibiotics, that will have no effect on the virus. And that's one of the reasons why we have antibiotic resistance, is prescribing drugs for a bacterial infection when you don't have a bacterial infection. So COVID-19 is teaching us the hard way that we have no treatment for new viruses. We don't have antiviral drugs for these, for the new coronavirus that we're seeing now. But now we do have vaccines, which can help us out. Okay, so if we look at the structure of a virus, it's fairly simple. It needs genetic material, and that material provides the information for how to build viruses, all the different component parts. And the genetic material is either DNA or RNA, and that's what's represented here by these squiggly lines. You want to protect that genetic information, so you enclose it in a protein shell, and this is called a capsid. And some viruses have an outer membrane called an envelope. And this is important because that's going to dictate which cell types they can infect and which organisms they can infect. Can I infect humans or not? And then which cells in a human or any other animals might I be able to infect? The other key component, about this envelope is that it's made up of fat particles, lipids. Well, this is important because if we take simple soap and we wash our hands properly, then you blow away that envelope and you kill the virus. That's why simple hand washing can be effective to prevent and deter the spread of an infection. You guys have already told me that you understand that viruses can't replicate by themselves. They have to infect a cell and use that host cell to manufacture more virus particles. Listed here are just a lot of common viruses or diseases caused by viruses. Some of them might be familiar with you. Rabies virus causes rabies. Herpes virus would cause a herpes infection. This virus, human immunodeficiency virus, actually causes this disease, acquired immune deficiency, whereas Zika virus causes a malformation of the brain called microcephaly. We're not always linked with how to name these viruses, but I'll tell you that there's more of a concerted effort going into this to make it simple to name them and simple to understand when you're naming a virus or a disease that you're talking about the same entity. So what happens when a virus infects a cell? So the virus has, on that outer envelope, it has something that might be able to stick to a cell. The virus can then enter the cell, and the genetic material within that virus hijacks the machinery inside the cell to make all of the components seen here to put new virus particles, I call them, together. They're assembled, and then they're released from the cell. Now you have a whole bunch of more viruses that can go infect more cells. Okay, so in this first part, the introduction to viruses, what I told you was there's just thousands of different types of viruses that we can find them throughout the ecosystem. And viruses are definitely different from bacteria, and it's important to know that distinction. And that it's the structure of those viral particles that indicate which cells can be infected.
Hi, excuse me.
Yep.
This is Steve.
Hi, Steve.
When you say using the word infected, it makes you think that viruses, I know that's a technical term, but it makes the connotation is that it's something negative that viruses are doing all the time. Is that the case, or are there positive and negative viruses?
Yeah, good question. Unlike bacteria, we have good and bad bacteria, and we can't live without bacteria. We need that, our microbiome. And viruses, in terms of naturally occurring viruses, there can be some good viruses. I don't know of any relationship between a virus and a human that's a positive relationship. But certainly, if you look at spiders or scorpions, they can have viruses that would indicate, they would change the biology of those organisms. So maybe skew them towards making more males rather than females when they reproduce. So if that's a good thing, okay, maybe. But I will tell you about some really cool, crazy things that scientists like me do with viruses that can be good for medicine and health. Any other questions? Okay. And feel free to jump in when you do have a question. All right, so that's your introduction to viruses. And now what I'm going to do is tell you about coronaviruses. So there are thousands and thousands and thousands of different types of coronaviruses. It's a really large family. The genetic material that they use is RNA rather than DNA. And if we look at this image here, this is showing 3 coronaviruses. And what you see are these little things sticking out of this virus, and it looks like a crown. This is how these viruses got their name. And if we were to look at a 3D image, that's what this image below depicts. And those crowns are these spiky things sticking out of the envelope of this virus. So typically, coronaviruses cause respiratory infections. And if that respiratory infection is an upper infection, in humans, it would be cold-like symptoms. Or a lower respiratory infection, it would be maybe pneumonia or bronchitis. But typically, coronaviruses don't really, out of the thousands, they don't often infect humans. But they're more widely spread in animals. And it could be cows or birds or bats or dogs or camels or whatever, you name the animal. So out of the thousands, there's only 7 coronaviruses that are known to infect humans. And out of that 7, there's only 3 seen here that have emerged within the last 2 decades that have caused serious illness and in some cases death. So if we look here, SARS-CoV stands for Severe Acute Respiratory Syndrome coronavirus. MERS is another coronavirus that can cause infection and death in humans. This is the Middle East Respiratory Syndrome coronavirus. And the coronavirus that we're currently dealing with in this pandemic is called SARS-CoV-2 because it's related to this first SARS it looks genetically similar to it. So it's number 2.
Just to
And
clarify the
yes,
question, this is David again. Just to understand, there are 7 known to infect humans, and in the last, say, 20 years, 3 of them has manifested themselves.
yep.
So is that suggest that as we go forward, this could become a bigger problem?
It could. It could, but there's good news, because every time we have an infection and one of these viruses kind of, causes a problem, we learn a lot. And that will become more apparent, I think, later in the talk. So it's never all bad and it's never all good. It's somewhere in between. And we're getting smarter as these infections are occurring. Okay, so I want to be clear on what happened. How do we name a virus and how do we name a disease? I gave you some examples previously, but there's actually this committee, the International Committee on the Taxonomy of Viruses, that names viruses. So there's a standardized format for classifying viruses. And this is important because if I'm talking about SARS coronavirus 2, I want to know that when I'm speaking to somebody else, we're talking about the same virus. And typically now, these viral names are based on the genetic makeup of the virus and how related they are to another virus. So SARS coronavirus 2 is related to SARS coronavirus, the first one. That's why it has a similar name, and then we just add a number to say it's the second one that we've found. Okay, so when you name diseases, that is not up to this committee. This is up to the World Health Organization. And we all know COVID-19, but maybe you didn't know where the name came from. It's coronavirus disease, that's COVID, and 19 means that it was first emerged in 2019. And it turns out that naming a virus can be kind of tricky because it could be confusing for people that cause them to avoid something that they shouldn't, or maybe it would have a negative connotation, as in, Middle Eastern respiratory syndrome. Maybe that's a negative tag attached to that region now. So even as recent as the last week, the World Health Organization is still kind of working out how to name these, but they have decided that the disease names shouldn't have a location within them. They shouldn't have names of people, occupation, animals, or types of food. So I told you that there are 3 coronaviruses that have caused widespread illness in humans. So here's SARS coronavirus that emerged in 2002. There were less than 800 deaths and about 8,000 infections. The Middle Eastern respiratory syndrome coronavirus, there's a little over 800 deaths and about 2,500 infections. So the key point here is look at the drastic difference between those 2 infections and our SARS coronavirus 2, which is what we're dealing with right now. This is a global pandemic, so it's in just under 200 countries as opposed to just a small fraction. Within the first 3 months, there were more than 1 million infections and greater than 50,000 deaths. So the data that I could find as of May 30th of this year for the US, there were more than 33 million infections and just under 600,000 deaths. And for Canada, it's about 1.3 million infections and 25,000 deaths. So the numbers, the sheer numbers that we're looking at are just completely crazy compared to the prior 2 coronavirus outbreaks. And I would mention here that of these 2,500 infections for the Middle Eastern respiratory syndrome coronavirus infection, there were only two infections in the US. Okay, so there's a lot of talk about where did these viruses come from? And the answer that I'm going to give you is based on scientific evidence. And essentially, what this graphic is showing you is the DNA sequence of coronaviruses and how they're related to other coronaviruses. So the data that's used is genomic data. That genetic material that's found in a virus, we can look at the viruses that we find in humans, and then we can look at viruses that we find in other animals and find out how related are they genetically. And we can do this over time, and we can look in different regions to try to figure out how is or was this virus transmitted. Through this type of evidence, it was found that the MERS actually originated from camels and then transferred to humans. And SARS-CoV-2 that we're dealing with now originated in, it's thought maybe bats or pangolins. Pangolins are like anteaters, and then transferred to humans. And importantly, there is nothing in this evidence that supports some of the conspiracy theories that arose saying that it was manufactured in somebody's lab. So when we look at viral transmission, most of the coronaviruses infect animals, and they don't often transfer to humans, but we know 7 times this has happened. So when an animal virus then begins to infect humans, this is called a zoonotic shift. And if that virus causes an infection, it's a zoonotic disease. So how does this happen? You need 2 things for this to happen. So the virus that causes COVID-19, it's thought that it initiated in a bat, and then it transferred from a bat to this pangolin. And then it undergoes changes to its genetic makeup that allowed it to transfer from pangolins to humans. And this is a working hypothesis based on the data that we have today. And as we learn, do more sequencing and learn more about it, we might be able to pinpoint it a little bit more precisely. And that's why you have the question marks here. But in order for this virus to transfer to humans, well, the humans have to be exposed to an animal carrying the virus, number 1. And then number 2, the virus has to be able to infect human cells. So how do these viruses infect our cells? Okay, so remember, here's a coronavirus, and you have these proteins coming out of the outer shell of this virus. These are called spike proteins. Do you guys hear that term in the US, spike proteins? Maybe some yes, some no. Okay, yep, okay. So it's these spike proteins found right here that allow the virus to bind to our cells and enter our cells. So the infection by SARS-CoV-2 is especially prevalent in airways because we breathe in air and the virus is in the air. So in order for the virus to be transferred from an animal to a human, there had to be mutations in the DNA or RNA, pardon me, the genetic material that make up these spike proteins that now make that spike protein for SARS-CoV-2 really good at binding onto cells and entering the cell. So what happens next after a virus then gets transported to humans? Well, you could have containment. And we're familiar with containment because there have been geographical restrictions. The border between Canada and the US has been closed for more than a year to just recreational travel. We can wear masks. We can wash our hands. We can socially distance. All of these can help to contain the virus. And then the other thing you want to do is enhance your immune response. And that's what these vaccines do. They ramp up our immune response to try to prevent the virus from infecting our cells. So by working on these 2 avenues, then you can reduce the number of new infections, and then the virus will be contained. It won't be able to be spread further. Well, another scenario that could occur is coexistence. So the virus infects humans, and then we transfer it to other humans, but it's still around. And there's unique features to those changes in the genetic material that allow that virus to stay and perhaps keep infecting. And we've heard about variants in the news. This is, we want to keep monitoring those because those tell us how this virus is evolving. And here we're looking at how fast is it evolving. So we need to sequence the genetic material of the variants to find out how did they change, how are they getting better at infecting cells, because that's going to help us create a plan to prevent that infection from happening. Okay, so what I told you about in this section on coronaviruses was that really out of the thousands and thousands of coronaviruses, they typically only infect humans, and only rarely do they transmit from animals to humans. There's only been 7 coronaviruses known to infect humans. 3 of them can cause serious illness and death. Typically, they cause respiratory infections. That the source of the virus that causes COVID-19, SARS coronavirus 2, it was a naturally occurring in nature. It wasn't man-made. And that we have measures to try to contain the virus, but we need to continually monitor across geographical regions and time points to see if the virus is changing.
Let's go back.
Okay, any
one-
questions?
Oh, here
Yep.
we good. I'm still, and maybe you don't know, what was particular about this virus that caused that animal to transmit it to a human? Could the animal have sneezed, or I just don't understand how that happened.
Okay, so if you have a dog and you love your dog, and I'm not saying this is transmitted by dogs, but let's say you have an animal and you're patting them and they lick your face, or yeah, they sneeze, or when you take your children, young children, to a petting zoo, you always want to wash their hands afterwards because these viruses are everywhere. Bacteria are everywhere. Some of them are okay, and some of them can be harmful. So they're actually quite easily spread. So the same way it would spread from an animal is the same way that it would spread in humans. And that's why we would wear a mask and stay apart particular distances, because viruses can't fly, they can't walk. So usually, yeah, it could be a sneeze or something like that, or talking that would transport the virus.
I clearly understand the human to human transmission. I guess what I'm trying to get at, if there was only 7 that have been known to infect humans, what happened with this particular one that apparently infected an anteater? And I know there's some people that love animals, but I can't see too many people snuggling up with an anteater.
So this comes down to culture, and you may have heard that the virus was thought to maybe originate in what's it called, a wet market in China, where they have different types of animals that they would eat, and they're not cooked. So you could transmit the virus in this wet market. So the conditions aren't what we would think of as hygienic. And then you have a lot of different animals in this wet market. So yeah, we're not typically snuggling anteaters and whatnot, but there are, depending upon your culture, times that you would come across animals that could transmit the disease.
Okay.
Any other questions?
Yeah, you've mentioned
Yeah.
a few times that the virus did not originate in a lab, and I know that's what we've been hearing. But recently, there's been a lot in the news about maybe we don't know as much as we needed to know about the origins of this of the virus. Can you speak to to what are the avenues of investigation that need to go forward, and what are the questions that aren't answered at this point?
So if typically the information that we're using now is based on genetic data, sequencing nucleotide by nucleotide, what the virus looks like. And the mutations that they found in SARS coronavirus 2 that allow it to infect human cells would not be the same mutations that a scientist would incorporate to make the virus more infectable. So all of our intelligence and our computer programs wouldn't have predicted the mutations that allowed it to infect more readily. We hadn't seen it before. And then typically, if you were going to manufacture a virus, you would use a backbone of an existing virus and then just add a few things into it. And we've worked with viruses for over 50 years. In 1987, I started working at the Hospital for Sick Children in Toronto in a lab that studied blood stem cells. And one of my jobs was to take a virus and take out the genetic material that we didn't want to create space so I could put a gene in that virus that I could then use the virus to infect cells to correct a genetic defect. So the other part of your question was what evidence do we need to be looking at? And we need to be continually doing this genetic surveillance of these viruses in animals and people over time in different regions, because what I'm telling you, remember those question marks? We think it came from bats and then to anteaters. This is our working hypothesis right now. And right now it's valid until you have information that suggests that it's not. So just keep monitoring. And you want to put I'm sure there are other surveillance mechanisms that the government would probably look into. Communications, we speak of the dark web and things like that, or people communicating that they want to try to do this, but that is a communication. It's not evidence. You still need the biological evidence.
I have some questions.
Yeah.
I actually have 3 questions.
Yeah.
Saving them up. The first one is, I had heard that bacteria are living things, but viruses are not living things. And I wonder if you could talk about that a little bit. Number 2 is, they're so small that 500 million of them could fit on the head of a pin. And yet you talk about manipulating them by inserting things into them. So you must have something that's a lot smaller than the virus itself. Like, what is that? And then number 3 is, you made a comment about, well, this one is not DNA, it's RNA. And that was supposed to mean something to me, but it really doesn't. So those 3 things. Thank you.
Okay, so I'm going to, hopefully I remember all of those, but the first one was, you've heard people say that viruses are not living. And it's one of the things that people remember about viruses, and this is my personal opinion. I think it's a useless piece of information, because whether they're living or not, they cause a lot of havoc. Number one, or number 2, maybe we need to change our definition of what it is to be living. So that was one point. So your next question was, I said DNA or RNA, and you weren't certain how to handle that information. Genetic material that gives the instructions to make all the virus parts comes in 2 different flavors. It's DNA or RNA. So it's not important except to know that it's the information that allows more viruses to be produced. And your second question, you're going to have to remind me what that was, please.
Yeah. How do you manipulate something that's so small? How do you get into the inside of something that's so small?
It's molecular biology. It's technological advancements and molecular biology. I don't see it. I know that it's there and I manipulate it, and then I can look at the biology of the outcome of that to know that I've manipulated it correctly. Or I take many more than 500 million viruses, and then I expand the material that I changed to try to detect it. Did I make the change that I wanted to? But yeah, we can, I think the wonderment here is that we can work on a really small scale and make micro manipulations that we can detect. And we you know, that was 1987 when I started that. So the technology has really changed, but it's kind of fascinating.
Yeah, so an electron microscope can see one virus at a time, but
Yep.
you don't manipulate one virus at a time. You manipulate a bulk of them and hope that some end up being manipulated. Oh,
I manipulate the genetic material, and then I package that into viruses through biology.
yeah, but not one at a time.
Not one at a time.
All right, good. Thank you.
Barbara, this is Charlie. I have a question or two if I could ask.
Sure, Charlie.
The first question I would ask is with the two very successful RNA vaccines, does this give us a leg up in the future for the mutations that can occur from COVID-19? I assume it does, and also to unknown variants of COVID coronaviruses. And the second question is, the way the mutants create themselves, do viruses do this at a more rapid rate than you would expect in the normal biological world, or is it just the way they do it?
So the answer to the first question is yes. It gives us a super leg up and reason for exceptional optimism that hopefully will become more apparent when I speak about vaccines. And the second part of your question, just remind me again, sorry. Charlie, the second part?
I'm unmuted, sorry. Their ability to replicate on a
Yes, thank
scale
you.
of
Yep.
rapidity, is it super fast, or is it normal for these viruses?
Yes, viruses don't do things faster than other organisms. It's to the best of my knowledge. It just comes down to random chance. So anytime that you're going to make a copy of genetic material, about 1 in a million of those nucleotides can be mutated. If it's advantageous for that virus, meaning, you know, maybe it could infect human cells more readily, then those viruses will be the ones that will be successful to keep replicating more. If it mutates and it makes it disadvantageous for that genetic change to infect human cells, those viruses won't propagate. But they're not mutating faster, as far as I know, than any other, the replication of any other genetic material, even in human cells.
Would you be talking about the vaccines in particular? There are different models out there. There's the Moderna, the Johnson & Johnson, and the Pfizer. Will you be talking about that?
Yes, I will. Yep.
Thank you. Okay.
Yeah. Will we move on to that right now? And then if you
I have a question. I was
Okay.
unmuted. Are you going to be also including the variants and how they seem to be in some places more than in other places, and how they affect us? And also, there's a book you probably know about called The Code Breaker by Walter Isaacson, and about the gene editing, and Jennifer Doudna. And it's fascinating. I haven't read the whole book, but it's the whole business of the RNA, the history of it, and how she does it, and what she does, and everything.
Yeah.
Really good.
Yeah. I agree. The technology is rapidly changing, and it's fascinating, and what we can do with it to help us out with infection and disease and regenerative medicine repair is exciting. And the variants, if I don't answer your question about that in the next part of the talk, please bring it up again, because it might make more sense after we go through the vaccines and vaccine development.
Okay.
Thank you.
Question?
Okay. Sorry?
Where do viruses fit in terms of evolution? The evolutionary tree, we always are exposed to that. So what can you say about evolution of viruses and bacteria?
Yeah, that I can't speak to off the top of my head. I'd have to look it up. Okay. All right, so let's talk about vaccines and vaccination. We probably don't have to tell this group why you need to vaccinate. Vaccines save lives. So for example, you're probably familiar with this machine. This is an iron lung. Most of the students that I teach have no idea what it is. Years ago, poliovirus was a big deal, and many people died and became paralyzed, and some of them had to be put into these machines to help them breathe. And through a robust vaccination program, many countries, including Canada, are now considered polio free. But we do have to be wary, because even though we vaccinate and we may not overtly see the disease, it doesn't mean that those viruses aren't still around. And in some cases, so here's an example in Ireland, where cases of measles drastically increased from 148 to about 1,200. And it was because there was a significant drop in the number of people that got immunized. So it says that there's a need for vaccination and a need for consistent vaccination. And the other thing that we have to think about is that vaccines protect everyone. And there are people that, because of medical reasons or because of their age, they can't become vaccinated. So it's up to other people to vaccinate to prevent the spread of in this case, a virus, to try to help everyone in the community. So the basics of vaccines. So unlike most medicines that would treat, or in maybe some cases even cure a disease, vaccines actually prevent a disease or infection. And they do this because they contain foreign germs. Now, these germs are either killed, or they're a weakened version of the germ, or it could just be germ parts. So a key feature of the vaccines is that they can't cause the disease. What they do, however, is stimulate your immune system, make it aware of a foreign germ, to stimulate a response, so that if you do see that germ out in nature, you will be protected from that through developing immunity. So when we say that the vaccines contain foreign germs, we call these antigens. Your body recognizes these antigens as foreign, and your immune system makes antibodies. These antibodies are targeted only for that antigen. So it's like a key in a lock. It'll only fit one lock and not fit the other ones. They're very specific. Important points are that the immune system has a memory. So if you see this antigen once, you build up antibodies. The next time you see it, you have a quicker response to defend yourself from that infection, because your immune system has a memory, and then you can rapidly destruct this disease-causing germ. Okay, so now let's take vaccines and speak specifically about SARS coronavirus 2. And the star of vaccines in this respect is the spike protein. So here's an envelope. This is a coronavirus, and you see these red spikes, the spike proteins coming out of the envelope. So these spikes are what the virus uses to bind to and then enter host cells. And this virus is very good at binding to human cells right now. So we also know the genetic makeup of this virus. And right here is a part of that genetic makeup coded in red. And that's to signify that these red spikes are being coded for by this gene. So we take that little piece of genetic material from that virus that codes for the spike protein. And that's what these vaccines are using to create immunity against COVID-19. Okay, so there are different types of vaccines. Basically, there are viral vector vaccines. And a vector just means it's a transport system. So if you want to get something inside a cell, think about an efficient way to get it in there. Well, take a virus that would infect a cell and use that as transportation. So it's viral vector vaccines. There's AstraZeneca, which, as far as I can tell so far, is not approved in the United States, but it is in several countries. And there are Johnson & Johnson. And these vaccines are based on DNA. It's not a big deal. The other two vaccines are based on messenger RNA, and these come from Pfizer BioNTech and Moderna. So what I'd like to do, I want to accentuate that the vaccines required the genetic sequence of this virus in order to be made. We needed to know the instructions for that spike protein, and that genetic sequence was available to the public 12 days after Wuhan declared a cluster of pneumonia. So it happened really fast. And so just in general, we've been using viral vectors since the 1970s, use them to study gene therapy or for biological research because they're just really handy tools. And here are just some of the publications that I was a part of when I was at Sick Children's Hospital, ranging from the early to late 90s. Using proof of concept, if you had a disease, we studied blood stem cells. If you had a disease where your immune system didn't work right, there was one gene that was mutated. So if you could transfer a good copy of that gene into stem cells that repopulate the blood, then you could fix that disease. That's part of what these publications were about. And if you study how blood normally gets formed, well, then you should study what goes wrong with blood some of the time. So cancer, we also studied leukemia. And we used viral vectors to take this, this fusion gene called TLS-ERG, used a virus to infect normal human blood cells, and then determined that this gene was one of the key players on one way to get leukemia. So we've used them for a long time. There are treatments, FDA-approved treatments for, not FDA, clinically approved treatments for Ebola and other treatments for HIV and Zika virus that are in clinical trials right now. So it's not a new technology. Okay, so let's look at how these viral vectors vaccines work. So this would be for the AstraZeneca or the Johnson & Johnson. So basically, you take the genetic sequence for these spike proteins. Remember, that's how the virus binds to and infects your cell. And we package them. Here's the DNA for the spike protein, and it's packaged in this little compartment called, this is the shell of an adenovirus. Remember when I said there's different types of viruses, and remember this one? Well, here it is. All they did was take the outside of the adenovirus because it's really good at infecting cells. So it can't make more adenoviruses. It's simply a vector, a transportation mechanism to infect cells, to protect this DNA, and then insert that DNA into human cells so they can make copies of the protein. So here's our adenoviral vector. It can bind to cell membranes, human cell membranes. It gets pulled into the cell, and the DNA gets transferred to the nucleus. In the nucleus, remember the end result is that we want to make a protein we want to make the viral spike protein. That's how our immune system is going to see that it's foreign and make antibodies to it. So on our travel to make protein, if we start with DNA, there's an intermediate molecule here called RNA, messenger RNA. So there's two ways to make protein. You can either start with messenger RNA and then make protein, or you could start with DNA that makes messenger RNA that then makes protein. So for the AstraZeneca vaccine and the Johnson & Johnson, they started with DNA. The DNA makes messenger RNA, and then the messenger RNA gets shuttled outside of the nucleus into the cytoplasm, where it's converted into protein that we see here. So this is, it's only making the spike protein of that virus, nothing else. It doesn't contain the entire genetics. Those proteins get put on the surface of this cell. So these are human cells that now have a viral spike protein on them. And your immune system comes around and says, hey, wait a minute, that's foreign. And it starts to make antibodies against the spike protein. So now, if you were to come across the SARS coronavirus 2, your system has antibodies for the spike protein, it binds to them, and now this virus can't bind to your cells and it can't infect your cells. Your immune system will also mark this virus, and in some cases, cells that have been infected with the virus, to destroy them. And then hopefully you can eradicate the virus. Okay, so there have been some questions, causes for concern. And one question that I saw a lot was, since adenoviral vectors are based on DNA, can they alter my DNA? And again, based on scientific data and decades of research, the answer is absolutely not. Adenoviruses, as they occur in nature, they have no ability to integrate into the DNA. There are some viruses that can, but these viruses cannot. And this is one of the reasons why there's such a great platform to start building and manufacturing vaccines from. These vaccines are also engineered so that they can't make more adenoviruses. They're just using this shell on the outside. And there's been many clinical trials for many different avenues of research that have shown these types of vaccines to be safe. Okay, so what I was telling you about in this first portion on the vaccines and vaccine development is that vaccines save lives and they prevent disease. What they do is allow your immune system to create antibodies, and then these antibodies can destroy the disease-causing germs. All of the vaccines that are available in the US are based on the spike protein, the genetic sequence of the spike protein for SARS-CoV-2, and that the viral vector vaccines that I just spoke of have been developed over many, many years. So we didn't just whip something up in the last year, but we're learning from decades of research. Okay, questions?
I have a
Yeah,
question. This is Richard G. If the cells, after the vaccine is put in, the cells have a memory, why is there a need for a booster?
Because your cells continually turn over. Okay, so you don't have, your cells only last for so long and then they die off. So you do need to have a booster just to refresh your immune system too, and really ramp up that that immune response so that it can be rapid when you do see the germ in real life.
This is Steve. I'm thinking about mutations And we have the polio vaccine, and you have the measles vaccine, and they seem to have worked over decades and decades. And now we're worried whether this vaccine may not work over 6 months, that there'll be a mutation and it won't work. Why haven't there been mutations that made the measles vaccine unusable? And why are there mutations that we think will make this unusable?
Yeah, so these are great questions, Steve. It comes down to infection rates. Remember, if every time you're going to copy the genetic material about 1 in a million times, you're going to have a mutation. So if we can get a vaccine, so for polio and measles and mumps, we've been really successful at our vaccination program. So the virus doesn't really have anywhere to go. If you have nowhere to go, you can't mutate, you can't become, a super virus. But we have seen people that now think, well, I don't want to have vaccines. So we have seen outbreaks of measles in certain communities, and certainly polio in other communities. And that's a concern, because that then can lead to the more infection you have, the greater the chances are that you'll have these variants. And so here in the United States, and let's bring it back to COVID-19, we're in the early stages, really, of the vaccination program. And it's wonderful that a large proportion of the population in the United States is vaccinated. But your neighbors to the north don't have that same level of vaccination. And they're pretty close, but we live in a global world. You just get on a plane and you can come into the US or Canada or wherever from a place that is a hot spot. So if you look at India, they're not able right now to really contain the infection, which gives you more and more and more opportunities to have mutations or changes in the genetic material that will make these variants. And I don't know if you use the term here, but in Canada, we call it variants of concern. And that's why you want to be testing people and animals at different times and in different regions to look at the sequence of that virus, to see how is it evolving, and are the vaccines that we have right now going to be able to counteract the evolution of the new viral variants.
I have more of a comment than a question.
Mm-hmm.
I had shared with Barbara before we began that I've had a kidney transplant during the pandemic, and there was no research about the effects of the vaccine on immunosuppressed people who've had transplants. The first study has just come out of Johns Hopkins University saying that 45% of us, the vaccines made no difference. And in the rest of us, the vaccines gave a limited antibody response. So just know that some of your neighbors are immunosuppressed, and we have to be very, very careful, because if we get the disease, it's very, very serious.
Exactly. Thank you for sharing that. And yeah, I too, I have a friend in Canada who's a heart transplant recipient. And yeah, I'm very aware of that community and trying to do everything that I can do to support them. Yeah. And getting vaccinated, people that are able to get vaccinated, like myself, that can help people that aren't able to.
I'm depending on the herd immunity of those around us. And unfortunately, even in New England, there are about 30% of people who have not been vaccinated.
Can you clarify a little bit of the concept of herd immunity? I know Kathy just mentioned it, and I think I understand what it means, but just kind of explain it a little bit, and again, explain why it's so important for those of us who can get vaccinated.
So it comes back a little bit to what Steve was asking also, is that, so if we have a population of people and a certain percentage of them are vaccinated, they're immunized, then the virus can't spread because your immune system can take care of the virus. So if the virus can't spread, it can't mutate. We don't have these variants. But if you, and I think there's debate over what percentage of people need to be vaccinated in order to reach herd immunity. It's somewhere in the 90s, but exactly where we don't know. So if you don't have enough people that are vaccinated, there's still a very good opportunity for the population to become infected with the virus. And the more infection you have, the greater the chances that there's a mutation that will occur in the virus. Will that mutation make it more infectable to other people? Maybe, but it doesn't have to. Will that mutation make the disease that's caused more severe? It could, but it doesn't have to. So it's kind of a lottery. So think of buying lottery tickets, and 1 in a million times you win. So if you get vaccinated, you don't have enough lottery tickets to reach that threshold to win, right? Or maybe you think of it the other way. If everyone is vaccinated, you do have all of this good fortune and the virus can't go anywhere. Did that answer your question?
Do you think
Yes.
that,
Oh,
go ahead.
I was just going to say yes, but how is that also impacted by, and I'll use the term natural immunity, someone who's had COVID and survived? Is there a difference in that kind of immunity versus one that results from a vaccine?
The immune system is the same and the concept is the same, but what might be different, like, you know, no two people are identical. Even if you have identical twins, they're not really identical. There could be underlying events that make you more susceptible or less susceptible. Could that you have maybe have other health conditions, or it could just be a something in your makeup that makes you more or less susceptible. But in the scenario that we were just speaking of, if not enough people get vaccinated and the virus can spread more, then you may not, even though somebody had COVID, they might not be immunized against the new version, the new variant.
Wow. Does the world need to have herd immunity, or just like the United States have herd immunity, or does it really need to be worldwide?
We need to think globally. You don't have to have herd immunity, but we do have to think about vaccinating globally, because we are a global society. We can easily be in a different part of the world very quickly and vice versa. So we need to think about the entire community. Okay, so I'm going to talk a little bit more about vaccines and vaccine development, and this is the last section for you guys that are wondering. So we just discussed how these viral vector vaccines were made, and now I want to talk about the messenger RNA vaccines, the ones that are made at Pfizer and Moderna. Okay, so these vaccines are based on RNA, and remember, what we really want to do is get to make protein, and all of the vaccines are based on the instructions to make the spike protein from SARS coronavirus 2. And one way that you can make that protein is to inject the messenger RNA for the protein into a cell and use the cell's machinery to make that protein. So the messenger RNA is just providing the instructions for the viral spike protein. The technology, conceptually, it's the same idea. They're just using a different molecule. And this is a picture of what the vaccine might look like. And these squiggles in here are the messenger RNA. And you'll see that there's a little baggie here, and then there's a baggie here. So messenger RNA is very unstable. It's very easily degraded. So you have to protect it. And they put it in this lipid membrane. And this outer lipid membrane also protects that payload package so that it can get, when you have an intramuscular injection, so that it can get into your cells. And this outside also helps this bind to the cells. So, okay, this you have this, it's called a lipid nanoparticle. It has something on the surface here that can bind to your cell, and this cell has now been vaccinated, and there's messenger RNA that's injected into that cell, and it uses the machinery in that cell to make the spike protein. The spike protein goes to the surface of the cell. Your immune system recognizes that it's foreign. It produces antibodies, and it protects you from the infection. So we came, this speed at which vaccines were available was the fastest that's ever occurred in history. So I wanted to look a little bit at how were these tested, and did they go through rigorous testing? So they didn't circumvent the rigor or the testing. The testing has strict regulations and authorities that overlook them. So for any clinical trial, and this graphic shows you something for drug development, but the same, the timeline might change a bit, but the same idea occurs for looking at a vaccine. So there are different phases to a clinical trial. There's phase 1, phase 2, and phase 3. And these phases are sequential, and each, you have to be successful at a previous phase in order to move on to the next phase. So essentially, phase 1 takes healthy individuals and injects in this case, the vaccine into them, and it looks to see how it affects them. You're not really looking for antibody production or does it prevent infection. You're just saying, is it safe to give people? And ruling out that there might be some adverse event that you would be otherwise unaware of. So, okay, if it's safe, then you move on to phase 2 clinical trials. This now is looking at the effectiveness of the drug. If you were to give this drug, does it deter infection in those individuals? And do they produce antibodies? And then if you're successful at phase 2, you can move on to phase 3. You're now looking at a broader, diverse group of people, larger and more diverse, to look at the ability of that vaccine to provide immunity. And so an important point for phase 2, and phase 3 is that these studies are called double-blind studies. That means that the medical professionals that are running the study, they don't know if people are getting the actual vaccine or if they're just getting a placebo. And the people in the study, they don't know whether they got the actual vaccine or the placebo. It wouldn't have the ability to make the spike protein. And then when the data comes back, it's all coded and you can decode it. So then you can separate it into these people actually got the vaccine and these people just got a pretend vaccine that wouldn't provide any immunity. So typically to get to phase 3 usually takes about 2 years for a vaccine, but in this case it took 6 months. So that was something that really helped to advance the rapid release of these vaccines to the public. And also, the vaccines were being mass manufactured before they knew the results of the clinical trials. And normally you wouldn't do that. And the reason that the manufacturers were able to do this is that governments gave them a lot of money to do this. Because normally you go through these phases and say, hey, I'm not going to spend a lot of money manufacturing this if it doesn't meet the requirements of the clinical trial. But this time, money was thrown at these companies to manufacture it because we just didn't have time to lose. And then the other development of these vaccines, I hope I've instilled in you that we've had previous coronavirus infections that we've learned from. Our technology, our ability to do sequencing and to build these viral vectors and to manufacture them has come so far where people really are, we have the intelligence and now we have the investment that people can see that these are great technologies. Okay, so in science, I'm a scientist. It's always important to record the experiment that you did rather than the experiment that you meant to do, because mistakes lead to some of the most fascinating discoveries. And here's an example for the AstraZeneca vaccine. So this vaccine requires 2 doses and you give them 28 days apart. And what they discovered, the people running the trial, was somebody made a mathematical error and one group of people only got a half first dose and then a full second dose, but it was supposed to be 2 full doses. That's what the clinical trial called for. But okay, so which, and now they could tease apart this data. They didn't know that they were getting this, but now they can tease it apart. So just think to yourself, which dose regimen do you think would provide better protection against COVID-19? So the half dose and then a full dose, or 2 full doses? So it turns out that this error worked in their favor, that 90%, if you were given a half dose in your first dose and a full dose in your second dose, it showed 90% effectiveness at deterring infection, compared to only 62% if you're given 2 full doses. So it's always important to be honest in science and to write down the experiment that you did, not the one that you thought you were supposed to do.
Do they understand the mechanism of why that was?
Not fully. And what they speculated, yeah, so I wouldn't have predicted that. What they speculated was this, that maybe if you gave a smaller first dose, that mimicked better what might happen in a natural occurrence. When you you wouldn't come across a whole bunch of virus, you come across maybe a smaller amount of virus. And then the second dose boosts up that immune response, but they don't really know. But certainly something to look into.
Is that also true of Pfizer and Moderna?
Is which true? The dosage?
That they actually gave a smaller dose for the dose 1 and a larger dose for dose 2?
No, and this wasn't planned in this clinical trial. Somebody made a mathematical error, and it turned out that this mistake led to an important discovery. But they haven't, so it hasn't been. It's something that would have to be tested in future clinical trials. Is this a better method? So we've had a lot of advantages with these messenger RNA and adenoviral vaccines that we have today for SARS coronavirus 2. They have a lot of advantages in that they're safe. You're not putting in a live virus. You're not putting in a whole virus. You're putting a little tiny piece in. So there's no risk of becoming infected with a vaccine. And the genetic material that is injected doesn't integrate into the DNA of the person being vaccinated. We know that they have efficacy, meaning that they do ramp up your immune response, and that the host, they integrate, they can make the spike protein in the host cells. We can make these vaccines really, really rapidly, and they're very versatile. So it's predicated on this, the genetic sequence. And so if it turns out that that spike protein and the sequence that we're using today becomes inefficient because there's a variant, then you take out that piece of DNA and you put in, or RNA, and you put in the sequence for the new variant, and then you start manufacturing that vaccine. So just like, you know, with the flu, we get a flu shot once a year because the variants change a bit. I can see us potentially getting into that scenario with SARS coronavirus too. And then you can apply this technology to any different target that you want. We only talked about the spike protein, but you could take other portions of that virus and make a vaccine against it. And they're starting to use this technology for things like cancer therapies and whatnot. So the future is very exciting for this, the technology and how quickly we can create vaccines for a variety of different uses.
I have a question.
Yeah.
Some of the people who don't want to get the vaccine say that they don't believe that there was enough research done.
Mhm.
And I don't know why they don't read information about it, but that's their reasoning. But it seems, based on what you're saying, that that's not true, that the research was adequate and proved. I mean, we can tell people are vaccinated and they're doing better.
Yes, exactly. And but the other point is that the choice of whether to be vaccinated or not is an individual choice, I suppose. I mean, some people have the choice and some people don't, as we've discussed. But for people that do have the choice, yeah, they're not being forced to take the vaccine. You know, there are certainly good reasons to become vaccinated. And yes, if you did have concerns, I like your strategy, Catherine. Let's just read about it and convince ourselves, right? But not everyone does that.
No, or they listen to the wrong people.
Yeah. So these vaccines that have been developed for SARS coronavirus 2, they're safe and they're effective. And as Catherine just said, we can see the effects right now. They did undergo very rigorous testing. The other thing that made the results so quickly of the testing was normally you would get to the end of a phase 3 of the clinical trial, and then and only then would you disseminate the information from the clinical trial to, in the United States, the Federal Drug Administration for approval. But in this case, what the clinical trials did was, as information came in, they disseminated the research results immediately. So countries all around the world could evaluate it on a rolling basis. And that also helped with the rapid approval of these vaccines. So, and messenger RNA-based vaccines and adenovirus-based vaccines, we've been developing the technology for years, and now we're just reaping the benefits of the many years of development and learning. We certainly had knowledge gains from the 2 prior infectious coronaviruses that caused human illness and death. And because of the technological advances and investments and just being smarter, what I just said about releasing data as soon as possible, that really made the rapidness of approval and dissemination of these vaccines unprecedented. Yeah.
It's boring.
So, I have a question, Barbara. This is Charlie.
Questions? Yeah, go for it.
In Great Britain, you mentioned that the AstraZeneca was 28 days apart, and we know that they made some conscious decision to skip second shots. What has been the results of that delay? And also, now that the India virus is in Great Britain, I understand there's some second guessing about that, maybe.
I don't think anyone has a playbook on what is the appropriate path forward through this pandemic. So I think you do your best. You can always criticize, well, you could do A or B, and somebody does A, and then somebody says, well, you should have done B. So case in point, also in Canada, they gave out first doses to more people rather than giving the first and then the second dose within a certain time frame, and they've extended the length of time from 28 days to, I want to say maybe 16 weeks.
Oh, wow.
So, I guess the idea is, would you like to have zero immunity, or would you like to have a large population have some immunity, and then you still have to have the protective measures? Like in Ontario, we're still in lockdown. You can't, you can go to the grocery store, you could go to the beer store and the liquor store, but you can't go out and buy clothes. You'd have to order it online. You can't have people over. You can't go visit anywhere. You can't sit inside a restaurant or outside of a restaurant. So there are other measures that you put in place to deter the spread of the infection. Because the other side of that coin, if you did take your vaccines and you said, I'm going to double dose as many people as I can, and then the rest of the people have to wait, then you have a very small cohort of people that are fully vaccinated and a very large cohort that have no additional immune protection from a vaccination, which could lead to an increase in variants. And which of those you choose at the end of the day, you have to look at the data that's available to you at that time. And certainly changes in behavior, restrictions, and freedoms are following that current data. So follow the data and make your best educated guess based on the data.
Hi, this is Steve. I've read about potential research that they hope will be able to stop one vaccine to stop all coronaviruses. How would you even do that when they differ from each other so much? Is there some unifying factor in coronaviruses besides the fact that they geometrically have a corona?
Mm-hmm. Yeah, so a blanket, I'm not sure how you would do that, but theoretically, I would follow the approach that they've done now, prevent it from infecting the cell. So look at that spike protein. And so you could, and this is where the genetics comes into play too. And somebody was asking about evolution, but if you look at evolution is really relatedness, right? If you take a whole bunch of coronaviruses, and we have all of those sequences from all of those animals, and look at the common areas in the spike protein, according to those sequences, then if you target your vaccine for that sequence, then you should be able to block all kinds of, a large proportion of the coronaviruses. Theoretically, that's how I would do it. Although I haven't read how they actually are thinking about it, but that makes sense to me.
Okay. Any last questions? We're just about running out of time. But I'd like to thank you very much, Professor. Fascinating topic, and I'd love to have you come back in person and maybe talk about bacteria.
Yeah, it'd be great. Thank you very much for having me, and everyone be safe. Have a great day. Thanks, guys.
Enjoyed it.